In this assignment, we're going to get into the methods section of Trammell et al. 2013. So before you read the methods section, it's important to understand that when you're reading a scientific paper, you most likely are not going to be able to understand 100% of what's written in the article, and that's okay. In terms of importance, the abstract, the introduction, and the discussion section are where most of the time that's where you're going to get most of the information that you need. Usually when you're reading the methods section and also the results section, you're just kind of skimming over it. The exception to that would be if you were specifically trying to replicate their study, like for example, maybe if you wanted to set up a kelp reconditioning system in your river, you might be reading this in extreme detail because you might care what size tote they used, for example. But for the most part, for us and just reading this paper, it's not really the most important information to us. So just keep that in mind. This is going to be probably too much information than we need to know, but there's someone out there in the world that would like to know that information, and it's there just in case they need it. And so when you're reading the method section, keep that in mind. Especially when we get down to the data analysis part, like this stuff. I don't even know what this means. I could probably dig into the paper and figure it out, but if I was just reading this paper just to read it, I would just breeze right over that. Not really super important to understand what's going on. Okay, so a couple terms before we get into actually reading it. One is they mentioned some hatchery strays in the methods section. And so hatchery strays are hatchery salmon that did not return to the hatchery. For some reason or another, they returned to a stream and ended up spawning with wild fish. Okay, so that's what hatchery strays are. They also refer to something called a spring freshet. That's just a spring flood. So especially in systems that are fed by snowmelt, so up in the mountains, eastern Washington, these areas have these really big spring floods because when temperature is warm in the spring, that snow starts to melt and that puts a lot of water in the creek. And what that does is it acts as a trigger. So juvenile salmon, for example, and steelhead kelp, so steelhead that have already reproduced, will be looking for that spring freshet basically as a timing trigger. RKM is also mentioned. That's just river kilometer. It's basically just how far upstream you are. So it starts at 0 at the mouth of the river. And so the higher the RKM is, the higher the river kilometer is, the further upstream you are. There's also a section in the method section about scale analysis. So scale analysis is a way that we can tell how old a fish is, and we can also track their entry from saltwater to freshwater. It's a lot like reading tree rings. So each year a salmon will have a different ring on their scale, which will tell us how old they are. But also there's some different patterns that the salmon lays down on their scale, whether they're in saltwater or freshwater. So they can also use scale analysis to look at if a fish has entered saltwater or if it's been in freshwater the whole time. And then lastly, the statistics part. Do not get bogged down in the details of what an ANOVA is versus a MANOVA versus t-test. You don't need to know that. Basically, all these tests are doing is telling you, are your groups different or are your groups the same? And so if you have, for example, case 1 here, where maybe this is, for example, your control group that you did nothing to, and maybe this is the group that you did kelp reconditioning with, if they were very different like this, we can say with confidence that, kelp reconditioning really did increase, in multiple spawning events. Whereas if you have something down here like case 2, there might be a little bit of a difference between the two groups, but there's also a lot of overlap. And this is not perfect data. We didn't sample every single kelp out there. We don't have 100% certainty. And so when you have a situation like this, where they overlap a lot, we're not certain enough to say that they're actually different. And so that's what these statistical tests are doing. Are they different enough to be really, really sure that the treatment, that the kelp reconditioning actually made a difference? Or are they so similar that we're really not sure if there actually was a difference or if it was just some error or coincidence in the data? And it's okay to skip over the math jargon. You don't need to understand all these statistics, and you shouldn't be focusing on understanding every little bit and piece because, again, it's not efficient. And for our purposes, that's not the main thing that we're interested in reading this paper.